Deciphering the structure and function of DNA is a remarkable scientific achievement. Here are the facts. DNA is a long-chain molecule made up of just 4 building blocks. Let's call them G, A, T, and C for short. In cells, the DNA takes the shape of a twisted ladder. The building blocks, called nucleotides, all have the same molecular structure: a phosphate group, a sugar, and a nitrogenous base. The nitrogen atoms are shown in green. The nitrogenous bases are a little different for each of the 4 nucleotides: T, G, C, and A. Along the sides of the DNA ladder, the nucleotides are bonded together with strong chemical bonds, but the cross pieces are held by bonds that are easily broken. In these cross-links, A's will only bond with T's, and G's only with C's. The reason for this exclusive bonding arrangement is that A's and T's have 2 bonding sites, whereas G's and C's have 3. When it's time to replicate, a protein starts the process by landing on a particular set of nucleotides called the replication origin. These start points are found all along the DNA strand. Next, enzymes break the chemical bonds that hold the 2 strands together, opening up the DNA, forming a replication fork. Special proteins, the green eggs, form temporary supports, preventing the freed strand from twisting up. An enzyme lays down a primer, which acts as a start signal for replicating the leading strand. Now, in comes one of the most important enzymes in life, DNA polymerase. This is the enzyme that builds the complementary strand by adding on nucleotides gleaned from the cell's stockpile of loose building blocks, A's with T's and T's with A's, G's with C's, and C's with G's, building a new double-stranded DNA molecule. But what happens to the other strand, called the lagging strand? Due to DNA's chemical makeup, the strands run in opposite directions. Phosphate sugar base in this direction, while the other strand is going base sugar phosphate. So, to replicate the lagging strand, a DNA polymerase enzyme must run in the opposite direction. It clamps onto the primer and replicates the strand back to the previously completed section. In this way, the lagging strand becomes a series of short sections that are then bonded together with the help of yet another enzyme. This occurs all along the DNA until there are 2 new perfectly copied DNA molecules.